Hi everybody, Dr. Ahles here. In this short video, we're going to explore the way that light waves are actually detected and how we send them through the brain for processing. So let's start with a discussion of the two kinds of cells that we find in the retina. Remember that the retina is the innermost layer of the eyeball, and this is the layer where we actually perceive light waves. There are two kinds of photoreceptors, or two kinds of visual cells that we find in the retina. These cells are called rods and cones. It's going to be very important that you are able to compare and contrast rods and cones. No, I'm not going to ask you to identify them in pictures, but I am going to ask you to describe how they're different. So let's talk about those differences between rods and cones. When we talk about rods, we would say that these are highly sensitive photoreceptors. When I say that a rod is highly sensitive, what I mean is that it will work even in low light. It can detect any of the light that is around you. That is in contrast to our cones. We would say that cones are less sensitive of the two types of photoreceptors. For a cone to function, we must have bright light. So in other words, it takes a lot to activate a cone. It doesn't take very much to activate a rod. And when we talk about how these different kinds of cells are activated, it comes down to a difference in the kind of protein they use to detect light. So rods use a protein called rhodopsin. Rhodopsin is different from the protein used by cones, called photopsin. These different kinds of proteins detect different kinds of light. So the rhodopsin protein only detects whether there is or is not light. Doesn't tell me anything about the color, it just tells me if it's there or not. This means that rods only see in black and white, whereas cones, because they have various different photopsin proteins, are able to help us see in color. So our first compare and contrast between rods and cones. Rods don't need much light. Cones need a lot. Second compare and contrast. Rods use rhodopsin and see in black and white, whereas cones use photopsin and they see in color. When we talk about using these different pigments to detect light, we have a range of light waves that are detected by each of these particular pigments. So let's start with the rhodopsin protein, which is the only kind of protein that I find in all of my rods and cones. Rhodopsin only exists as this one range, detecting things in this one range of colors. So starting in blue or in purple, ranging up into blue, some green, a little bit down in the yellow, orange, red area, but not very much. Because there's only one protein, one kind of rhodopsin, either we've activated it or we have not activated it. That tells me, do I see something or do I not see something? Whereas in cones, we have three different photopsin proteins. Those three different photopsin proteins each are activated by a slightly different range of light waves. So notice that some of the cones in our eyes have the photopsin protein that's more sensitive in the blue range. It can detect light from about purple up through green. But then we have another cone that we call the green cone. This can detect light waves a little bit in purple, mostly blue, green, over into the yellow, orange, red range. And our final cone detects even farther to the right. So really it picks up in green, yellow, orange, and red. By having three different photopsin proteins that each detect slightly different wavelengths of light, cones can add together all the messages they're getting to figure out exactly what color they're seeing. So if you're seeing a color green, like this green right here, my green cones would be highly activated, my red cones would be kind of activated, and my blue cones would barely be activated. Your brain will add together all of the different activation levels of your cones to allow you to see in color. Interestingly, when we talk about seeing in color, there are some individuals in our population who may not be able to see all colors as clearly. One of the most common forms of color blindness is red-green color blindness. The inability of a person to see the difference between a green color, like we see on the outside of this picture, versus the red colors that are there in the middle. Color blindness, whether it's red-green or whether it affects other colors, has to do with these photopsin proteins. If they're made incorrectly, I can't detect light waves at those wavelengths. When we're talking about the location of rods and cones, remember that we had that area called the fovea centralis. This was in the very back of the eyeball in the retina. The fovea centralis is the part of the retina where we have the most cones. So this is the location where we have a high abundance of all of those cone cells. Remember, cones see in color, but cones need a lot of light to be activated. As we start going farther and farther away from the fovea centralis, we start to have fewer and fewer of those cones and a lot more of the rods. So when we're considering the locations of rods and cones, remember that cones are very centralized, rods are more on the periphery, they're more on the outside. This is an excellent comparison table from your textbook to summarize what we've said about rods versus cones. Remember that rods don't see in color, cones do. Cones are low sensitivity, meaning they need lots of light, where rods can focus in dim light. Remember that cones are what we find in the very center of your vision, the fovea centralis, whereas rods will fill up most of the peripheral parts of your retina, most of the parts on the side. One thing we hadn't mentioned, but we'll mention it now, is how numerous these types of cells are. You should know that there are many, many more rods in your retina than there are cones. For every cone, we've got about 20 rods that are also working in the background. So cones are less common, but they help us to see in color. When we look at the way that light waves travel through the retina, it's sometimes said that the retina is kind of evolutionarily backwards. Here's what I mean by that. We see a retina with 3 different layers of cells in it. We'll talk about these different layers of cells. But when light comes into the eyeball, it starts down here and it has to travel through all of these cells to get down to the bottom where the rods and the cones live. Rods and cones are the only cells in the retina that can actually perceive light. My other cells, like my bipolar cells or my ganglion cells, they can't perceive the light waves that pass them. So light starts on the outer part of the retina and has to travel all the way through the retina to get to the rods and the cones. Once the rods and the cones have detected those light waves, they'll send that information to cells like bipolar cells that we see here, or horizontal cells that we see here. The bipolar cell layer is my first layer where I summarize information. So imagine that I asked every single one of you, what's your favorite kind of pizza? Each of you, like a rod or a cone, would have an answer. Well, if I go to the bipolar cell level of seeing, it'd be like me asking, actually, I don't want to know every person's favorite. I want to know the favorite across your entire row of classmates. So we summarize our information and decide as a row what kind of pizza we like best. But then I tell you, actually, I need each of my sections in our big lecture hall to tell me the best kind of pizza. We're summarizing our summary of our individual choices. That's the way that vision works. Photoreceptor layer, the rods and cones, very sensitive, but we summarize their information in the bipolar cell layer, and we summarize that information in the ganglion cell layer as well. Ultimately, the ganglion cell layer is the layer that's going to help us build that optic nerve that goes out the back of the eyeball. So, the fovea centralis, we've said this before, this is the part of your eyeball where you see the most clearly. The fovea centralis sees very clearly for three main reasons. The first reason is that this is where light naturally focuses. The fovea centralis is right behind that pupil. The lens bends that light, and a lot of it gets focused on the fovea centralis. The next reason though, is that the fovea centralis is the location where we have the most cones. So the highest concentration or the highest density of my light detecting cells is found in this location. There's many, many cones at this location. And the other reason that we see so clearly in the fovea centralis is because this is the one part of the retina where we don't have those bipolar cells summarizing things, and we don't have those ganglion cells summarizing things. None of my light's going to get blocked or refracted by those types of cells because literally at the fovea, they're not there. Light goes directly to my rods and cones. So keep in mind there are several reasons that we see really well at the fovea centralis. But remember, the fovea centralis, with all of its cones, only works in bright light. Cones only work when there's a lot of light. So the very center of your field of vision, it's great for focusing. We've got nothing to obstruct the light waves, but you better have a lot of light. Otherwise, it's not going to work. The final thing I want to mention in the process of vision is talking about where we send our detected sensations to be processed in the brain. Attached to the back of the eyeball is cranial nerve number 2, the optic nerve. When we talk about the optic nerve, this is going to carry those signals out of the retina, out of the eyeball, back through the thalamus in the middle of the brain. Ultimately, we perceive visual sensations in the occipital lobe, the very backmost lobe of the brain. And remember, we said that about 50% of all the neurons in your cerebrum are involved in the process of vision. So from collecting light at your eyeballs to sending it through that optic nerve that crisscrosses back into the thalamus, back into the occipital lobe, lots and lots of neurons involved, helping you with the process of vision.